So our next speaker is going to be Jiyoung Ahn. Jiyoung Ahn is Associate Professor in the Department of Population Health and Environmental Health at NYU Medical Center. Her research focus is usually trying to understand the causes and prevention of cancer in human populations. And she's been very, very active in cancer research, a lot of NIH grants, publications, awards, that kind of stuff. So she's very qualified to give this lecture. So today she's going to connect with what our second speaker just introduced, which is the microbiome. She's going to talk about the human microbiome and diet and how that influences cancer health disparities. Thank you for coming, Dr. Jiyoung.
Hello, thank you for having me today. I want to talk about human microbiota today. Human body hosts trillions of microbiota and create ecosystem. Until last 10 years ago, we were not able to study them because majority of bacteria don't grow outside of human body. So it was really impossible to study them. Recent development of genomic sequencing technology widely opened microbiome area to study comprehensively, including culturables and non-culturable bacteria, and we can accurately quantify them. So totality of microbiota is so-called human microbiome. As a population-based research scientist, my research focus on the established relationship of human microbiome and oral digestive cancer disparity. And unlike small mouse group, 5 mice, 10 mice, humans are more complex. So we think it's important to look at diet and lifestyle factors and how these factors influence on human microbiome and how this microbiome the diet and cancer relationship is mediated through microbiome. Of course, we do expect some independent effect of diet and lifestyle factors for cancer. My overall goal is to identify human microbiota associated with the cancer. I think this is important because identification of bacteria may lead to personalized prevention by microbiota and lifestyle modification. So this is my talk agenda. First part, I'm going to focus on fairly recent data on diet, gut microbiota, and colon cancer in New York City Asian Americans. Later part of talk will focus on smoking, oral microbiota, and head and neck cancer risk. I become interested in racial disparity in Asian American colon cancer because, in general, in native country, colon cancer rate is low. But US Asian American immigrants immigrate to US, their colon cancer incident rate increase, although their rate is still lower than other races. So why is that? I become curious about this. New York City is racially, ethnically diverse. According to New York City Community Health Survey, 43% are foreign-born, including me, and 25% use non-English at home. In particular, Asian Americans are fastest growing racial group in the New York City, account for 13% in New York City, compared with average 4% in US overall. This picture shows each dot shows at least 50 people reside in the census area. You do see how Asians reside fairly a lot, particularly in Chinatown and Queens Flushing area. I was really fortunate to be able to develop New York City based on multi-ethnic cohort, so-called family study, food and microbiome longitudinal investigation. We recruited multi-ethnic population healthy volunteers, particularly we targeted Asians, Hispanics, Black, and white. We recruited them through churches, communities, local community center, and local health clinics. Currently, we have enrolled about 5,000 individuals, and we plan to go to achieve 10,000. And this involves prospective follow-up for health outcomes. We ask them to provide stool and oral samples for microbiome assay, and all participants completed demographic and lifestyle questionnaire, as well as diet questionnaire. All materials were translated in multi-languages. So in the beginning, I said microbiome is assayed by genomic sequencing. Let me just briefly talk about how we did it. 16S RNA gene sequencing is a fairly well-established method for microbiome. And why 16S RNA? This is because all prokaryotes carry this ribosomal component. And it serves as a molecular clock to identify bacterial phylogeny. And this bacterial gene here is fairly small and simple. It contains highly variable regions and also highly constant regions. So we designed the primers to capture these taxonomically identifiable V3, V4 gene regions. And this is a simple overview. From the stool sample, we added an individual barcode and amplified those gene regions, and we sequenced them. What we get is highly paralleling about 500 millions, 500 base pair sequencing read. Based on that, we cluster them based on sequence similarities, over 97%. So each unit is a so-called OTU, operational taxon unit, which is a pseudo-species. So with this OTU, we build a phylogenetic tree based on genomic phylogenic information. And so each individuals we can classify from phylum, class, order, family, genus, and species level. So each one, you will get the information, each individual's bacteria profile per person, per sample. So in the beginning, I said I'm a population-based scientist. This is what we learned over years. So across the whole population, in human population, we tend to share common structure. So here, so everyone in this room, I can tell, everyone will carry Firmicutes, Bacteroidetes, Proteobacteria, Actinobacteria, Fusobacterium. These are all common. We share these common members. However, each individual, we do have each individual variability per each content. So it's probably you cannot read, but this is like 1,000 individuals bacteria profile in genus level. You do see somewhat variations. So my bacteria content, overall microbiome content, is not necessary with Dr. Lee's microbiome profile. So we do have some individuality. So this is inter-individual variability confer disease susceptibility. And what are the determinants of this inter-individual variability? So before I talk about family study, I want to go back to several years. In 2013, we conducted a case control study of colon cancer and microbiome. We recruited 100 colon cancer patients at the clinic, and we also recruited age, sex, race matched health non-cancer control group. We simply compared their microbiome. And this is what we learned. Cancer groups, so this clodogram is a nice way to summary, and then cancer groups shown the red. Cancer groups are 2.5 times more likely to carry Fusobacterium compared to control group. And the second point is Clostridia, which is commensal bacteria, was significantly depleted in cancer patients compared with the control group. Other groups, basic scientists, reported that Clostridia play an important role fermenting diet fiber to butyrate, and which is known as an anti-carcinogenic component. Later, one of my fellow, Dr. Lin, showed that high fiber consumers tend to have high Clostridia abundance in healthy population. So let's go back to the family study. This is what we learned. So about 1,000, we sequenced about 1,000 those people who participated early participant in family study. We sequenced the microbiome. In particular, Dr. Peters, my postdoc fellow, she received the pilot funding from postdoc funding to looking at the Asians and white gut microbiome differences and acculturation. So this PCOA plot is a nice summary of showing the microbiome differences. So each dot is called a Johnson-Shannon Diversity Index, which is a summary index. I'm not going to tell you all the formula, but in a just short word, each dot summarizes each phylogenetic profile, and each two dots are close. That means their microbiome phylogenetic distance is close. They do have a similar microbiome profile. If dots are apart, that means their microbiome profiles are very different, overall structure. So what we've seen is a striking difference between Asians, shown by blue dots, and then non-Hispanic whites, shown in red dots. And now, I apologize for this small font. So now, you want to know what specific bacteria contribute for these gut microbiome differences. What we learned is particularly Clostridia abundance was particularly different between Asians and whites. So we found the colon cancer related bacteria. And obviously, it's known for fiber metabolizing bacteria when you compare the fruit and vegetable intake in Asians and whites, and Asians tend to eat more fruits and vegetables. Well, so this is predetermined with this. But what about, is it really like a race, or would it be more, what about without just knowing the determinant? Why don't you just to analyze this data differently? She did unsupervised clustering analysis, and then we found there are 4 separate distinct microbiome groups. So here, So here the first blue reached in Korean population, shown in the blue here. And this one, the second interesting group is pink group, and primarily non-Hispanic whites. And green and purple, they are at mixed group. So again, it's another way of visualization. And we measured comprehensive diet intake with food frequency questionnaire. And we do see that Korean-rich clusters are associated with high seafood, tofu, soups, cooked grain, fruits and vegetables, versus this white-rich group is strongly related to high cured meat, poultry, fats, and oils and cheese. So this is still ongoing analysis, fairly new, working on process project, but we realized this race is not probably black and white. There is an acculturation process going on. So we are currently looking at what are the bigger determinants of this diet acculturation. So we are looking at social, cultural, neighborhood impact in this New York City resident diet acculturation and microbiome effect and possibly cancer effect. So our tentative summary is East Asian immigrants increase colon cancer risk after moving to US. Lower gut Clostridia may be associated with higher risk for colon cancer. Asians have a different microbiome, at least partially explained by diet and bacteria. And we are still working on understanding diet acculturation among Asians. Now I'm going to moving on to my second talk, smoking, oral microbiota, and head and neck cancer risk. A while ago, several years ago, I showed that people with moderate and severe periodontal disease, which is inflammatory gum oral disease, is 2 times more likely to develop and die from upper GI cancers, cancers including head and neck and esophagus cancer. This is based on secondary data analysis of CDC and Ahnz data. Why is it? So my hypothesis is this could be due to something oral bacteria. Subsequently, we obtained the NCI funding. We were able to study oral microbiome and these upper GI cancers. The way we approached was we used a prospective study. This is in collaboration with the NCI and American Cancer Society. When I was in fellow at NCI 15 years ago, NCI developed this population cohort. And this NCI and ACS recruited 140,000 healthy individuals. And they collected, when they were healthy, they collected mouthwash samples. That time, mouthwash samples were designed to get human DNA, but now we are using for the microbiome study. So those 140,000 healthy individuals were followed up over 10 to 15 years. During this follow-up, some people developed the cancer, majority of people never have developed the cancer. So we selected those prospectively developed 130 head and neck cancer cases and 106 esophagus cancer cases. And we also selected age, sex, race matched, cohort nested controls, those who never developed cancers. And this is important because we can study bacteria profile when they are healthy, and we can see which bacteria profile is associated with the subsequent development of these cancers. So we completed Assay. This is what we have found for head and neck cancer. From this is a beta-proteobacteria group. We learned that people who carry more proteobacteria in their mouth associate with 30% less likely to develop head and neck cancer subsequently. And this is the first study to show the direct link of oral bacteria and head and neck cancer. At the same time, we also showed that this same bacteria is inversely related to esophagus cancer development, although effect size is a little smaller than head and neck cancer. So now you might wonder what this bacteria does. And we didn't test the function directly, but we created the inferred metagenome profile. In that way, we can see which genes each bacteria carries, and we can group them by CAG gene functional pathway. So we learned that these beta-proteobacteria groups play an important role in xenobiotic biodegradation pathways, including chlorhexidine, nitrotoluene, styrene, toluene degradation. You might wonder, so what? And these are important smoking carcinogens. Now, so then we decided that, well, that if that is true, then this relationship would be more pronounced in the high carcinogen environment. So we divided our group, former, never, current smokers, and we evaluated the relationship of proteobacteria and head and neck cancer. As expected, we found the inverse relationship of proteobacteria and head and neck cancer were more pronounced in current and former smokers. So this is a summary. Greater oral beta-proteobacteria abundance decreased risk of head and neck and esophagus cancers. Beta-proteobacteria may involve in metabolizing carcinogens. And this may lead for targeted prevention for beta-proteobacteria, particularly in smokers, while encouraging them to stop smoking. So today I just talked about, my talk was focused on cancer development area, but there are also important areas we are working on is cancer progression. Those people who developed and will be treated, whether these bacteria play a role involves both treatment response and progression. For example, melanoma, immunotherapy, those people who treated, whether microbiome predictors for response to therapy. We are actively collaborating with other area of scientists. For example, we identify bacteria. These bacteria need to be tested in basic science for the mechanism. And another group of collaborators is actively working on identification of developing probiotics and specific antibiotics. And lastly, we are actively collaborating with method development experts, like statistics and laboratory. So we are in the very exciting area times, and we just begin to understand about human bacteria and cancer relationship. So why this is important? And identification of bacteria may help to learn about the causes of cancers and may help us to identify people at high risk for cancers. And this may lead to ways to prevent cancer by tailored microbial and diet lifestyle modifications. So this is truly based on teamwork, and we have great collaborators at NYU, CDC, ACS, and NCI, and I am grateful for funding support from NCI for make this project happen. Thank you.